Hey guys, so today I want to talk a bit about my honors thesis project that I did, and that's the project that got me interested into microbiology. So let's get started. So my project was like any other project for any undergraduate student. It was you chose your supervisor and you did basically a one-year project on any biological or science-related research. And for me, it was on hand dryers and bacteria. The primary objective of our project was to understand the microbial or bacterial ecology found in these hand dryers and washrooms, and to understand the numbers that are produced, but also the types of bacteria, and finally the antibiotic resistance coming from these bacteria from hand dryers itself. Now, the type of hand dryers we tested on were the vertical hand dryers, where you place your hands vertically and the air is hitting your hands perpendicularly. I'm not going to say the brand name because I do not want to get in trouble, but you guys kind of get the concept of which hand dryers I'm talking about. Those were the ones that we tested on, and here are the results. The project was very simplistic. It utilized very basic microbiological techniques, such as isolating bacteria, dilution series, a little bit of susceptibility testing using Kirby-Bauer discs. And so overall, a very simplistic project, especially compared to the other projects that were being done that year. But I felt was the most interesting project, personally for me. The quantities of bacteria produced from these hand dryers really varied, depending on what sort of method you did. And for us, in our project, we used different particular methods of quantification. Specifically, we used swabs, we used our hands, and then we used air samples, and we had exposed agar plates. When we looked at the numbers of bacteria being produced on the surfaces of our hand and on the surfaces of the hand dryer, what we noticed was that on the surfaces of our hands, the hand dryers were not emitting much bacteria. And whether you used a hand dryer or a paper towel, there was not really much difference in terms of the numbers of bacteria, and in fact there were very few bacteria being produced. However, when you took the swab samples and you swabbed the surfaces of the hand dryers, the inner surface of the hand dryers were containing a lot more bacteria.
Damn!
We're talking close to 100,000 colonies or CFUs per mil, to close to a million CFUs per mil. And so, what was really important to note there was that the surfaces of the hand dryers were had high contamination of the potential bacteria that we will notice. But when we tested the air samples by exposing the agar plates to the hand dryers and exposing the agar plates to the washroom in general, what we noticed was that the agar plates that were exposed to the hand dryers were producing bacteria numbers of close to 100 colonies on the plate.
Oh, hell no.
Whereas the bacteria produced from just exposure to the washroom didn't really produce much. In fact, less than 1 colony was produced on those plates. So it was quite interesting to see that while it may not be emitting bacteria onto the surfaces of our hands, it was still emitting bacteria onto the surfaces of our agar plates. And so what could be potentially a problem is that we might be inhaling aerosolized bacteria, even though it may not be in contact with the surfaces of our hands. So something to just kind of keep in mind about. Now, because of the COVID pandemic that we had, the project was kind of halted. So we couldn't really do any molecular analysis in the identification of the bacteria that we collected.
Nice.
And some of our antibiotic resistance testing was also limited as well because of that. We couldn't test all the samples, but of the samples that we tested, about half of them were resistant to at least one antimicrobial.
Nice.
Which was quite interesting to see because antimicrobial resistance is quite an issue in today's world. And so we have to understand what is leading antimicrobial resistance and why is it occurring in these bacteria. Now, from my understanding, fecal bacteria coming from our intestine or from poop, generally is higher in terms of resistance. So all in all, it was a great project to work on, and it got me interested in microbiology in general, and hence why I'm here today doing my master's thesis in microbiology in general. Just to let you guys know, this has not been published. This is not a published paper, but it was for my honors thesis, so it was something that I held in high regard. But yeah, hopefully it kind of was interesting for you guys. Anyways, guys, please like, follow, and comment, and yeah, I look forward to doing the next video. Cheers, guys.